The University of Oslo's 2020 International Genetically Engineered Machine team have been working hard with our project Sal Coli, where our small interdisciplinary group and all of our work presented here is theoretical. Our work comprises a theoretical synthetic biology plan and a detection system. Aquaculture is an important part of Norway's economy, but it is unfortunately plagued with inefficiencies. This includes fish loss caused by disease, and our project focuses on minimizing this loss. We have made theoretical plans to engineer E. coli to produce salinomycin, a precursor to an antiparasitic compound, to help combat a disease called amoebic gill disease. To complement this, we have been working on an early detection system to diagnose different fish diseases. Our detection system focuses on observing collective behavior to detect changes caused by individuals getting sick. To start the design phase, we have adapted a model of schooling behavior, implemented a classifier, and a visualization tool.
Fish farming is the practice of raising fish for commercial gain, usually food, on land or at sea. This is the most common form of aquaculture. Aquaculture is an important industry in Norway and accounts for a large proportion of our exports. This is also a rapidly growing market worldwide, with a projected value of $376 billion by 2025. While fish consumption is growing rapidly worldwide, overfishing is unfortunately increasing too. The practice of fish farming can potentially alleviate this problem by reducing the amount of wild capture fishing. An important factor for any production is efficiency. Currently, there are large losses in aquaculture. In Norway, the loss in terms of number of fish is between 15 to 20%, of which deaths account for a large majority. Among the causes of death are parasitic diseases such as sea lice infestation and amoebic gill disease. Increasing the efficiency is a step in the right direction when it comes to aquaculture's toll on ecosystems and, of course, fish welfare. AGD is an increasing problem in Norway. The parasite causes respiratory distress, suffering, and can lead to death. If AGD is left untreated, some aquaculture facilities have reported up to 80% mortality. Treatments for AGD exist, such as freshwater bath that kills amoeba by osmotic shock. In addition to that, AGD can be treated with hydrogen peroxide. However, none of these treatments are perfect, and often many rounds of treatment are required depending on severity. The diagnosis of this infection is unfortunately rather slow and relies on gill score based on visual inspection, which only detects severe symptoms and often confuses AGD for other diseases. Here are the components of what we call a detection system. Early detection is generally beneficial for all ailments and often reduces the amount of treatment required. This is also the case with AGD. We wish to achieve early detection of AGD by creating a system which detects behavioral changes. We know that changes of behavior is an important symptom in many fish diseases, and it does also occur as the earliest symptom in some. The reported behavioral indicators are quite extreme, something that makes it an obvious sign to humans. But if the development of these symptoms are continuous, then an automated detection system might pick it up sooner. This would also be true if disease symptoms develop in discrete steps for only a limited number of fish at once, since these fish would be hard to observe by a human. While there are many projects working on tracking individual fish and their swimming patterns, our unique approach aims to look at collective behaviors.
Less abrasive treatment to the current industry standard. The current treatments of AGD mentioned earlier causes a great deal of stress and potential harm. With our project, we want to offer an alternative to these treatments. A first step in this direction is to transfer the gene cluster for production of salinomycin, an antiparasitic compound, into E. coli. We hope that producing this compound using synthetic biology methods would complement our detection system and provide a less abrasive treatment to the current industry standard. Our first step would be to clone the Luxar family transcriptional regulator, SLNR, into E. coli cells. How would we reach that goal? Our plan would be to clone the salinomycin-producing gene cluster from Streptomyces albus into E. coli. Since salinomycin targets gram-positive bacteria, the gram-negative E. coli is a good candidate for salinomycin production. Our first steps would be to isolate SLNR from Streptomyces albus gDNA. We would then clone it into a pBed His expression vector and transform it into chemically competent E. coli cells. We would then aim to verify successful transformation via colony PCR and subsequently sequencing of transformants. To verify functional insert, we would perform expression purification of protein of interest and electrophoretic mobility shift assay.
As mentioned earlier, we have been looking at collective behavior instead of individual behavior. We went with this idea for the following reasons. First, collective behavior might amplify individual behavioral changes, and measurements on collective behavior might be easier to obtain. Second, we have not found anything similar in literature when it comes to fish disease, making our approach rather unique. During the design phase of our detection system, it is beneficial to have a computer model with which we can run simulations. This would allow us to explore different ideas on the implementation side, such as different sensors and classification schemes. The best solution for our classification scheme would probably be to use a machine learning approach, for which our model could generate practically endless data. In our project, we did not have time to create what we call an environment model, but this should be possible for others in the future. After researching modeling of collective behavior in fish, we found that schooling is the most common. We decided to create our own version of a previous model of this phenomenon. Extending the model we found in literature to 3 dimensions, this model now has a number of interesting parameters, including how much a fish values its own direction, allowing us to, for example, represent less reactive fish. With this model, the fishes attempt to stay at an interval distance from each other and move in the same direction by the use of 3 mechanisms: attraction, orientation, and repulsion. If fish are within the radius of repulsion, they will try to move away from each other. In the radius of orientation, the fish try to move in the same direction, while fish within the radius of attraction are attracted towards each other. After implementing this model, we wanted to see if we could detect the difference between 2 simulations with different parameters, given a simple metric. Here is the metric we used. If we could train a classifier to tell the difference between 2 states with very little information, while it is hard to see for humans, then this might also be applicable in the real world. Before we get into the results, we will introduce quickly one of the main concepts behind statistical classification. Classification is the process of assigning data to a category or class. In statistical classification, this is done by a classifier, where data is represented by feature vectors. Here x1 and x2 are features, and the space their Cartesian product spans out is referred to as feature space. These features could, for example, be weight and length. One could expect that different types of fish, for example, have different combinations of vectors of weight and length, and therefore occupy different locations in feature space. Each point here is a feature vector, where blue and green belong to 2 different classes. The end goal of a classifier is to find a decision boundary, here the red line. This allows us to classify new data by knowing which side of the line it falls in feature space. The same principle is also used by the classifier we implemented, a neural net. With machine learning, we do classification by what is called supervised learning, where beforehand we have a set of data with an assigned class for training. When we input some of this data consisting of feature vectors, the classifier will give some output. Since we know the class in the training set, we have a preferred output, say 0 for blue and 1 for green, defining what is called a loss function, a common example being the square difference between output and preferred output. We can define functions called optimizers, which attempt to minimize this loss function. An optimizer could, for example, be gradient descent, where we simply calculate the gradient at a point on the surface created by the loss function, and go in the opposite direction. The smart thing here is that the human does not need to know exactly how the classifier tells these two apart. One can almost apply this concept indiscriminately. In our case, we can easily create training data thanks to our model of fish schooling and train a classifier without knowing how it tells the classes apart. The same principle is also used by the classifier we implemented, a neural net. Accuracy for saying that all data belongs to that class. To evaluate our classifier, we use the metrics given here. Accuracy is the ratio of right classifications to all samples. Precision and recall tells us how biased the classifier is. If the set of data we are working on mostly contains one class, we would get a high accuracy for saying that all data belongs to that class. In the start of our simulations, there is no order. The system of fishes then undergo a phase transition to an ordered school system. After this phase transition, the fish are organized. For example, they could be migrating in a common direction. The most natural part of our simulation to classify is the steady state. At this point, the fish are actually schooling. But there is another phase, the transient one, that turns out to also be very interesting. We have found that it's much easier to classify the fish if one also observes this phase transition, as this part of our simulation is apparently more sensitive to the parameter changes we introduced. In our model, we have a migratory solution. In other words, the steady state is that most or all fish move together in a defined direction. The parameter choice is arbitrary, but we have found a baseline set of parameters which we call healthy. The other set is less reactive and have less affinity to orient and move towards each other. These we call sick. We take 50 samples of the metric given earlier, evenly distributed through the part of the simulation we are interested in, using 1600 feature vectors, of which 160 are for testing. We train and evaluate our classifier. The part of the simulation which was steady state was determined visually and was found to be between 30 and 100% of the simulation. With this setup, we routinely get an accuracy of over 65%. In the example shown below, we had a good run, getting an accuracy of 71.25%. These changes are caused by differences in initialization of the neural network, as the initial weights are always random in the beginning. Here we sample between 0 and 75% of the simulation. We then have the same interval size, but additional information contained in the phase transition. While the accuracy here is also variable, we routinely see it go over 74%. These differences in accuracy might be a good indication that it would be the same case with real fish, and that also in the real world there would be extra information contained in the phase transition. The phase transition here is a transition between two different global behaviors. In our case, we go from not schooling to schooling. In the real world, it might be interesting to look at other phase transition, such as disruptions caused by feeding. The exact characteristic of these phase transitions is not known by us, only by the black box classifier. For example, it might be the time it takes to form a school. While our accuracy might not be perfect, it is not of great concern. This because in the real world, one could measure and evaluate many times, and if the number of sick classifications is high, then one could contact a veterinarian or follow some procedure. It makes sense that in the early phases of a disease, there is little information to indicate its presence. Therefore, early classification would come with some unreliability.
As demonstrated in the videos, observing differences between healthy and sick is very hard for the human eye. Therefore, we believe that an automated system like ours might pick up these changes much earlier than humans. We have created a set of interconnected software pieces that allow us to simulate, classify, and visualize fish behavior. Our modeling code is extendable, and we are convinced that it would be possible to add new and more complex behaviors. The whole setup is relatively easy to use, though it requires compilers, as Julia is a compiled language. All parameters can be added in a separate text file. A user can explore the model of fish schooling by simply changing values in the said text file and hitting play. Visualization is a very powerful tool that allows users to observe fish in a lifelike manner. Lifelike visualization allows for model validation by human experts. One could then more safely implement experimental models and have some indication that the models are useful without needing to run experiments straight away. Human practices have been very important for us, especially integrated human practices. The discussion we had with experts significantly altered the path we took. We also reached out to public and asked them for their opinions on synthetic biology and gave interviews on 2 different media. For our survey, we created a questionnaire and managed to gather data during the corona pandemic and produced some interesting results from the responses. We asked 100 people to fill out our survey in Oslo. In the survey, it became evident that most people have no knowledge of synthetic biology, but are generally supportive of research done in this field.
We believe our software can be used further by researchers and the aquaculture industry. It allows for powerful visualization, which can be used for expert human evaluation of model performance. While developing the project further, it would be beneficial to create the environment part of the model. This would allow for the design phase of the physical side of the detection system, while also giving the ability to refine the classification scheme based on more exact input data generated by the physical implementations proposed. Our approach might also be used directly on a fish farm that has cameras, by estimating some collective metric and training a classifier on the gathered data. The classification scheme can be extended significantly. An interesting approach here would be to first tailor the model to a real fish farm by use of the environment model. Then one could set up a system of neural nets specializing in different symptoms of disease and try to verify that this works directly on real-world data.